Hello, our topic today is on the development of the brain, non-critical periods, and like other topics that we're discussing, please remember to take a look at those thought questions as well as those videotapes that have been um, assigned that you should be looking at. And in addition to the reading that you're doing, it's important that you keep up with that. So that you can better answer those thought questions that you'll be responsible for. So this is an important area of study in the field of behavioral neuroscience, and the reason why it has become so critically important is because it really helps us a great deal in terms of understanding different neurodevelopmental disorders. One of the topics that we'll be exploring will be autism, for example, which is a neurodevelopmental disorder. So we really need to know about different features of brain development that ultimately are going to help us to answer key questions about what various behavior disorders are related to, especially the neurodevelopmental disorders like autism, as well as other disorders on the autism spectrum. So one of the things that we talk about is this the neural pathways, the neural connections that are being laid down during early life. So if you take a look at this figure that you see on the bottom of the slide, you can see we're talking about prenatal life here, fetal life, and the first year of life, and then the first few years of life up to the teen years. And one of the things that we know is that when we take a look at the pathways that are important for processing of sensory information, vision and hearing, for example, those pathways are being laid down very early in life. I mean, take a look at what you're seeing here in terms of when this is beginning. It's beginning during the first beginning during later months of fetal life, and then extending into the first year of life. And then again, a lot of this progress in terms of those pathways is taking place during very early life. Take a look at language. That's the blue curve that you see here. Language also is developing relatively early in terms of those neural pathways that are critical. And then, higher cognitive functions, that's in the red that you see here. Those are things that are, those neural pathways are really accelerating in terms of their development during the first year of life. And then beyond that. So again, this is just to give you a little bit of a timeline. First, it's those sensory pathways, then language, and then the higher cognitive functions. So when we take a look then, there's really two important findings that you need to be aware of. First of all, early stages of brain development are really crucial for normal development later on in life. A lot of information in the field of behavioral neuroscience points in that direction. And secondly, there are chemical distortions in the brain that if they do occur during early development, they can cause a lot of problems in terms of the development of behavior. And again, you're going to be seeing a lot of evidence of this as we move along in the field of behavioral neuroscience. Again, we'll be taking a look at again, things like autism and attention deficit disorder, for example. So it's it's important that we understand this sequential pattern that is occurring in terms of the development of these pathways that are crucial for normal behavioral development. So just some examples of disturbances that can occur during pregnancy. Certainly we've known about radiation exposure for a long time. When you take a look at what happened, for example, when the bomb was dropped on Hiroshima, when you take a look at what happened at Chernobyl, again, that's in some of the videotapes that you're going to be watching. that are important for this particular area. Radiation exposure is known to have very profound effects in terms of behavioral development. And indeed, we know, quite a bit about that now. We know, for example, that the amount of time that you're exposed to that, the distance, how close or how far you are from radiation, and whether or not there's any shielding that's involved. Those are three really important factors that are going to determine the degree to which you may show impairments in terms of brain development. Alcohol exposure is another important area of disturbances that occur in the brain during pregnancy. In this case, we know a lot about what's called the fetal alcohol syndrome, and how alcohol passes into the fetus by way of the placenta. And we know a lot now about this particular disorder, this particular syndrome. So these are just a couple of examples of this kind of disturbances that can occur during pregnancy and the impact that it can have. So when we take a look at a little bit more depth in terms of fetal alcohol syndrome, this was initially noted in mothers who drank heavily during pregnancy. And it's really marked by, a lot of different characteristics. But some of the more important ones are ones that I have listed here. Certainly, this impulsiveness, the inability really of someone to inhibit certain emotional behaviors. Difficulty in terms of maintaining attention. You see varying degrees of mental retardation, motor problems, facial abnormalities. These are all pretty common characteristics of the fetal alcohol syndrome child. And again, here's just a better representation of some of those facial characteristics that you see. Typically, these kids have a relatively small head, low nasal bridge, these epicanthal folds that you see here that oftentimes gives the appearance of a child that is oriental in appearance. Relatively short nose, thin upper lip, an underdeveloped jaw, a very smooth philtrum, that is the upper lip that you see here, a flat mid-face, short eyelid openings. Again, these are all characteristics of the fetal alcohol syndrome child. We have a number of good models for studying this now. One is utilizes the mouse. And if you take a look at this slide, you see some striking similarities in terms of a child with fetal alcohol syndrome and mice, young mice that have been exposed prenatally to alcohol. And again, a lot of the same characteristics, small nose that you see here. These short, relatively short fissures that you see here in the eye, very similar in terms of the mouse, relatively small head. And again, this is, compare this to a normal mouse and then take a look at the alcohol exposed mouse, one that's exposed to this during early life, early prenatal life. And again, thin upper lip and the long philtrum that you see here, again, very similar to the human. So this has served as a good model for exploring the impact of this alcohol exposure during prenatal life. So again, some more of these behavioral and physical features. Again, here's some of these classic kinds of symptoms that you see in a fetal alcohol child. Their physical growth is relatively slow. Before and after birth, there's reports of difficulties in terms of visual processing as well as auditory processing, poor coordination, intellectual impairment, again, this delayed development syndrome, learning disorders, short attention span, hyperactivity, Again, this lack of impulse control seems to be something that's very characteristic of the fetal alcohol child. There's some reports of heart defects. So again, these are all, some of the behavioral outcomes that you see with fetal alcohol exposure. When you take a look at what is happening in terms of neural function, there's a lot of studies now that indicate that the dendrites of neural cells of children with fetal alcohol are short. They have very few branches. We also know that there's a suppression that occurs in terms of certain neurotransmitters like glutamate and elevated release of neurotransmitters like GABA. We also know that the important role that neurotropins are playing. There seems to be less stimulation that is occurring of neurons in the central nervous system, and there's this premature cell loss, cell death that is occurring, something that we refer to as apoptosis. Take a look at other kinds of exposures that do lead to that is prenatal exposures to things like cocaine, for example, during pregnancy. Children of mothers who use cocaine, there's a number of different deficits that occur. There's decreases that occur in terms of language skills. There's decreases that occur in terms of IQ scores. There's impairment that occurs in terms of hearing. So again, these are all outcomes of exposure, in this case, to an illicit drug. A growing area of study is that of what is called endocrine disruptors. And if you take a look at the disrupting chemicals that are out there, things like pesticides, some ingredients in household products, solvents, various industrial ingredients, pharmaceuticals, metals, these are all known to have a disruptive effect upon the endocrine system, the reproductive system in mammals. There's a lot of research that points in that direction. Some of that research has been done by a scientist at the University of Missouri, by Fred Vomsaal, who has been looking at a chemical, bisphenol, which has been used in plastics for a very long period of time. And you can see Dr. Vomsaal here with some of these plastic products that have been made from this particular chemical. Things like baby bottles, for example, are made with this particular chemical. So his research really has shown in literally hundreds of studies now that this particular chemical, bisphenol, acts a lot like the hormone estrogen. And when estrogen levels are high, there are a number of different outcomes, not the least of which is the change that occurs in reproductive ability. So this is something that's very concerning. And indeed is something that scientists have been exploring, and that pharmaceutical companies and environmental protection agencies have been at loggerheads, really, in terms of trying to come to some kind of resolution as to whether or not the use of this particular chemical should be suspended. It has been suspended in some countries, but not in other countries. So it's something that's a very controversial area right now. Take a look at some of these other impacts that can occur from, in this case, smoking during pregnancy. Children of mothers who smoke during pregnancy show risk for a number of different characteristics in their offspring, like low birth weight, for example, sudden infant death syndrome, attention deficit hyperactivity disorder, intellectual deficits, deficits in terms of the immune system. So again, this is another case of how disrupting what is happening neurodevelopmentally, in this case by way of smoking, can have a very destabilizing effect in terms of the development of the brain. So an important concept here is the one that we call plasticity of the brain. And it's important to really understand some of these key concepts, essential concepts. When we say plasticity, what we're saying is that the brain is changing, and it's changing throughout our lifetime. And certainly there's an awful lot of change that's occurring during early life, but there's also changes that are occurring during later life. So indeed, this concept of plasticity, that is of the constant change that is taking place in the brain throughout the lifetime of the individual, is crucial in this area. So the development of the brain, we know, is due to both experience with the environment as well as physical maturation. But the rapidity of that development that occurs during early life is profound. The prefrontal cortical area is developing very rapidly between 7 and 12 months postnatally. For example, if you take a look at the work that was done by the famous developmental psychologist Jean Piaget on a phenomenon that's called object permanence. Take a look at that work and what we now know about the development of the brain. It becomes very interesting. Piaget talked about this concept of object permanence. That is to say, with a young child, if an object is put in front of them, they reach for it, they enjoy playing for it, but if all of a sudden now that object is that they preferred is placed behind, in this case, just a piece of cardboard, it's out of sight, out of mind. So an infant at 9 months of age, they're not going to reach for that object any longer. It's been hidden. It's out of sight, out of mind. But give them a few months, and with further development of the prefrontal cortical area, they're going to reach for an object that they have seen hidden. So again, this is strongly associated with what is happening in the development of the prefrontal cortical area of the brain. So we can explain object permanence then on the basis of this. If you take a look at early brain development, this is a figure here which is showing the development of what we call the neural tube. Central nervous system begins to form when the embryo is only about 2 weeks old, and the dorsal surface begins to thicken, and the neural tube, there's fluid that fills this cavity that you see right here. The forward end starts to enlarge and ultimately differentiates into the hindbrain, the midbrain, and the forebrain. The rest of the neural tube ultimately becomes the spinal cord, as you see right here. When we explore the stages of brain development, this is what it looks like at 3 weeks of life. Again, here's the forebrain, here's the midbrain, here's the hindbrain, parts of the spinal cord. This is at 3 weeks. At 7 weeks of age, again, take a look now. Forebrain is developing more, as is the midbrain and the hindbrain. The cranial nerves are starting to develop. Then at 11 weeks, you can see further development of the forebrain. And especially the hindbrain that you see right here. Again, at the time of birth, again, you see this very accelerated development of the forebrain, and what looks to be almost adult-like in terms of its appearance. when you take a look at the growth of the human brain, and I think this is a really excellent chart to look at right here, you're seeing very rapid development of the brain, growth of the brain here during prenatal life, and then on up into the first year and a half, 2 years of age. Then that growth starts to slow down, as you see here. So at birth, the brain weighs about 350 grams. The first year of life, about 1,000 grams. And then in adulthood, it's going to be about 1,200 to 1,400 grams. And one of the things that we're going to learn in one of our later lectures is there's development that's occurring in the brain during adolescence, something that we didn't really know about until fairly recently. And a lot of that development, or the lack of full development of the adult brain during the adolescence period, may explain some of the behavioral differences that you see during the period of adolescence. That's something that we'll get into in one of our future lectures.